Hello friends, we are meeting again. Today we'll be summarizing the discussion regarding excretory products and their elimination. Excretory products are nothing but the waste, metabolic waste, which are to be eliminated outside the body of our organism. This excretory products includes ammonia, urea, uric acid, carbon dioxide, guanine, and creatinine. Such excreted materials have to be differentiated on the basis of which organism excretes more in the major amount, and this is actually deciding the mode of excretion. We'll begin with the mode of excretion with the photographic explanation of uh, different materials like proteins. They are broken down to amino acids. Nucleic acid, they are broken down to nitrogenous bases. They all together form amino groups, and from the amino groups, the agents which will the organisms which will be excreting ammonia majorly, including the aquatic animals and many fishes, they are following ammonotelism. Then those who are excreting urea in as a major excretory product, including mammals, which includes human beings also. Then most amphibians, sharks, they are following ureotelism. And the birds, insects, and many reptiles, land snails, they excrete uric acid majorly. So they follow uricotelism. So depending on this, there are different modes of excretion. And shifting towards the ureotelism, as human beings, we follow this type of excretion, mode of excretion. So there is a urine formation through which this urea is actually excreted outside the body. So how this urine is formed and which organs are associated, we'll see in the coming slides, like including the anatomical or organ systems of urinary system in the body of a human being, which is including the kidney right and left, located at the door posterior position of the body, then the ureter, which lies medially, and finally the bladder, which stores the urine collected from both the kidneys, which lies anteriorly to our body, then the urethra. The sizes of urethra depends differentially on the basis of the organism. Like if the female body is there, the size of urethra is smaller. If male body is there, the size of urethra is larger. So in the kidney, there are structural and functional unit named as nephron. Nephron is the structural and functional unit of the kidney. And there are number of nephrons in this kidney. If you see the TS of kidney, there are number of nephrons located. And this number of nephrons, they accumulate, they form and collect the urine, which is transferred through the ureter and then reservoirly act as a reservoir. The bladder, it acts, and from the bladder, it is eliminated through the urethra. We'll see this is the structure of nephron. In this nephron, the upper cup-like shape, which is called as Malpighian corpuscle, or also known as Bowman's capsule or glomerulus capsule. This capsule is actually located in the medulla region of the kidney, and the different tubules lying beneath this corpuscle, they are coming in the in the cortex region of the kidney. And from this, the collecting duct, it takes all the urine which is formed from different nephrons from these branches, and it takes towards the ureter. So what is happening in this is there is afferent and efferent arteriole which are passing through this glomerular glomerular corpuscle, in which they act as a point of filtration. How this point of glomerular filtration takes place, we'll see here. In this corpuscle, the blood is flowing from the afferent arteriole. It takes the blood, comes over here. It is purified or filtered, we can say. The waste products is getting filtered and enters into the proximal convoluted tubule, which is the PCT, and the remaining blood is given out from the efferent arteriole. We can see in this diagram that the artery size of afferent and efferent is different. The afferent arteriole is larger in size and efferent is smaller. This is this anatomical behavior of this corpuscle makes it to generate a pressure due to which the filtration takes place. In this kidney, about 125 ml per minute filtrate in men is accumulated, and 105 ml per minute filtrate in women is accumulated. This is the average I'm talking about, and this amount equates to be the volume of 180 liters per day in men and 150 liters per day in women. So there is all so much amount of filtrate actually comes depending on this change in diameter which generates the pressure. There is a glomerular hydrostatic pressure over here, and this pressure creates the differences through which there is transfer of the filtrate in the lower areas, that is the tubules. There is a net filtration pressure which comes by a calculation, like the glomerular hydrostatic pressure, which is coming from this region, minus the capsular hydrostatic pressure, which is coming from this region, plus the blood colloid osmotic pressure, which comes from the blood vessels. It is all total coming to be the glomerular hydrostatic pressure over here is about 55 mmHg. The corpuscular hydrostatic pressure is about 15 mmHg, and the blood colloidal osmotic pressure is about 30 mmHg. So altogether, by this addition and subtraction, there is overall 10 millimeter Hg of pressure which is generated. Net filtration pressure, which is outside or filtrate going outside, is about 10 mmHg. With the pressure of 10 mmHg, it is liberated outside. In the outside, it means into the tubule from the corpuscle. So moving back to this diagram, from this corpuscle at a rate of pressure of 10 mmHg, the filtrate is entering in the PCT. From the PCT, there is changes occurs in which the salt and the different material and the sodium ions, the water molecules, they are absorbed and reabsorbed and selected. And this whole process of urine formation takes place from this PCT, then in the loop of Henle, then going towards the DCT. From various exchange, absorption and reabsorption, which is taking place at this tubular level, a final product of urine is actually coming over here in the collecting duct, and from the collecting duct, it is liberated towards the ureter, and from the ureter into the kidney, into the urinary bladder, and then lastly to the urethra. So this is how exactly the urine formation is taking place in the nephron. And there are more details of changes in pressure. Apart from changes in pressure, how minutely from where the sodium is getting absorbed, from where the water is secreted, what is happening, how the tonicity is changing, where the hypotonicity or isotonicity is there. We have to give it a separate time so that detailed urine formation can be studied. But for the summarized explanation, this much is enough for all of us, that how and where the changes are actually taking place. And then this is a summary of the primary urine, how it is passing, the filtratus passes the PCT, which reabsorb the useful substances from the primary urine. Partial absorption of water and sodium ions takes place from Henle's loop. This is important things to remember, from where the things are getting absorbed and reabsorbed. Sodium ions and water are getting reabsorbed under the influence of hormone in DCT. Urea, uric acid, creatinine are secreted by the tubule, and lastly the urine is formed. So this is how urine formation takes place in the human system. So the excretory products and formation of those collection of those waste products into the into the nephron from the different uh, corpuscles which are collecting and filtrating the blood, and then from this urine is actually formed, and this urine is then liberated outside the body depending on the changes. And this functioning of kidney is actually also associated with the major important function, which is termed to be as renin-angiotensin-aldosterone system, that is the RAS. I'll give the will give a brief explanation of what exactly the RAS is. We'll be thankful to the Encyclopedia Britannica for this wonderful picture, due to which we are going to understand what RAS is actually doing in our body. From the kidney in the juxtaglomerular apparatus, when renin is released from the kidney, it will react with the angiotensinogen, which is coming from the liver. The angiotensinogen and the renin, they together act to form angiotensin 1. This renin is actually liberated on account of the blood pressure changes. When there is a need, at that time the renin is released from the kidney. So the angiotensinogen and the renin, they together convert to form angiotensin 1. The angiotensin 1 needs to be get converted to angiotensin 2. And how it will take place? It will take place to the very important enzyme, that is the angiotensin converting enzyme, which is released from the lungs. The angiotensin converting enzyme is liberated and it converts angiotensin 1 to angiotensin 2. It is also termed as ACE. It is a very important part of RAS, which is also a target of many drugs which are used in hypertension and other cardiovascular problems. And then angiotensin 2, it acts on the adrenal gland and stimulate the release of aldosterone. This aldosterone is then also act on the kidneys to stimulate the reabsorption of sodium chloride, that is the salt, and the water. And also the angiotensin 2, which is coming from this step, it is acting directly on blood vessels, which is stimulating vasoconstriction. That means the narrowing of blood vessels, which will cause increase in blood pressure. So the RAS at the end, when it produces angiotensin 2 and aldosterone triggering, it will cause firstly the vasoconstriction, and that means it will increase the blood pressure. And the aldosterone which will be formed, it will be acting as a reabsorption of water and salt in the kidney. So this is how the RAS is also acting. So and this is a summary of the total, what we can say, excretory product system, and the formulation of how the RAS is actually acting, and how this every process is liberated, like the urine formation and then the reticulating acting system. So with this, I will thank you to the audience for this patient listening, and catch you later in some more interesting system of our body. See you till that time. Bye-bye.